Trinity is a professor at the University of Minnesota. She's interested in the microbiology and biogeochemistry of all sorts of cool and unusual environments. A few years ago, we were both interested in the microbial mats and anoxygenic photosynthesis in these mats in underwater sinkholes. Trinity is still into that. I've moved on, unfortunately, from this very cool topic. Trinity is also interested in studying hot springs in Yellowstone and in the microbiology of glaciers. I think that's what she's mainly going to be talking about today. And so I give you Trinity.
That loud noise is not my computer. Yeah, okay. Thank you, Sebastian, and for the invite. It's so fun to do these in person. Again, it's very, very exciting to travel again and to have conversations with people in 3D. I should also mention that Sebastian and I usually meet up, or at least once have met up in the Bahamas. So maybe we should figure out a way, especially in to escape the Minnesota winters, to hang out at some sinkholes again. Okay, so I sent the super ambiguous abstract, so I was feeling not non-committal at the time of the invite about what I wanted to talk about. And so sometimes I promised to talk about Proterozoic and some other stuff, but I'm not going to do that. I'm going to talk about mainly glaciers today. Sometimes I title this talk The Geomicrobiology of Mid-Latitude Glaciers. Other times I title it The Past, Present, and Future of Pink Snow, or the lack thereof of a future of pink snow, potentially. And so that's what I'm going to talk to you about today. These are some of the folks that have done or are still doing work in the lab, and I just like to highlight them before I get too far into my talk. The work that I'll highlight today was done mainly by PhD student Taylor Price, PhD student at Purdue, actually, Jordan Miller, who recently defended Seth Bill as a postdoc in the lab, who's also moved on but did a bunch of the analysis that I'll show. And then where is he? Chris. Here is a original Washington person who has moved to, oh, sorry, Oregon. He is Oregonian, so he has recently joined the lab as a PhD student and will continue some of his work. And then we get money and we work with a lot of different folks that help sort of with different aspects of the research that I'm going to talk about today. You can't see it here, but I had this wonderful undergrad design this project. So we call ourselves the Fringe Lab because we sort of, we work on microbes that are on the fridge of life, whether that be really cold or really hot. And the lab thought it would be more fun to just be the ham lab. And so we have about 1,000 of these deli stickers that you can get for, I don't know, $1.99. On Amazon, so we are the ham lab in the deli case. Okay, so what does the lab do? We study microbial physiology and biogeochemical cycling, and we do this over space and time. And so we can think about space as here in this room, the distance between you and I. We can also think about the space in terms of geochemistry or geology. So there's many different ways in which we think about microbes across space. And then we also think about them across time. And so we're really trying to link some of the things that we see in extant environments to early Earth and potentially other worlds. So we think about things sort of at the cellular scale. So we don't study a lot of model organisms, but sometimes we study isolates and think about what they're doing in terms of physiology, how they evolved. We think a lot about how microbes live in communities. This is this lovely map from a sinkhole that you won't get to hear about today. But we think a lot about how sort of different phototrophs or different organisms within this system work together in a community. And then we try to link that to sort of larger scale processes in the Earth system. And so that's sort of where what we do in the lab is connecting across these different scales and thinking about space and time. What do I want to tell you about today? In terms of astrobiology, I'm going to talk a little bit about snow and ice ecosystems. And what do we study? What am I going to tell you about today? I'm going to tell you about the role of snow and ice in supporting downstream ecosystems. This is really not a very biology relevant story yet, so it's a little bit of a stretch for me. But really what we're thinking about is how snow and ice systems deliver nutrients, microbes, and meltwater downstream. So we think about these glaciers, or we do in my lab on these high alpine, or in the case of Oregon and Washington on these stratovolcanoes. These systems are not islands. They host their own diversity, but they also impact downstream ecosystems. And so if we're thinking about either early Earth conditions, snowball Earth, or potential for microbes or life on other planets, we're not just supporting, glaciers are not just supporting life there at the snow and ice. They're sort of delivering a lot of things downstream, and so potentially supporting a larger ecosystem. We also think a lot about peak snow and how peak snow may have contributed to past glacial interglacial cycles, and certainly are contributing to deglaciation today. So I'll talk a little bit about the ecophysiology of microbes on snow and ice. And really what we're interested in here is what controls their distribution and their abundance. And so thinking about how they're how they may have contributed to biogeochemical cycling through snowball Earth or other past glacial interglacial oscillations, what controls their distribution and how abundant they are. And then another pet project, or one thing we want to think about, is how microbes thrive on snow and ice seasonally. Even if we're thinking about Earth during past glacial interglacial oscillations, there were still some amount of seasons. And these communities are ephemeral, so they're there in the melt season when liquid water is available for processes. On snow and ice, anyway. On this type of snow and ice, not I'm ignoring marine ice systems for this. All right, so what am I going to tell you? I'm going to tell you that I'm going to come back to your great state and the state of Oregon soon and climb around in these mountains and look for snow, for big snow. But I'm also going to tell you that glacial melt contributes to mountain block recharge. Again, this is sort of a hydrology story, but we're interested in how alpine glaciers or mountain glaciers impact the systems around them, both near and far. And then I'm going to tell you two things about how snow microbes get nutrients. So the bottom line here is snow and ice is host to a lot of different microbes, diverse range of microbes. But from our observations on snow, supraglacial snow at least, these organisms still depend on delivery of external nutrients. And so that has implications for thinking about what the sort of distribution of these microbes might have been throughout Earth's history. All right. So I've sort of mentioned this already, and I'll show you some evidence for this, that there's periods of glaciation. Deglaciation are common throughout Earth's history. We're living through one of these now. There's evidence for glaciation on early Mars, and there's potential for life on icy worlds. So it's just what we're doing in my lab is asking basic questions about how microbes might live on snow and ice to sort of tell us how this might have happened in the past or on other worlds. Mid-latitude glaciers are also key components of local and global watersheds. I probably don't have to tell you that in this room, but I do have to remind folks when I'm talking to them in Minnesota. Last week I was talking to people at Baylor in Texas, and elsewhere, when people aren't sort of faced with or see glaciers or snowfields on a regular basis, we might forget that their contribution is substantial. Nearly 1/6 of the world's population depends on snow and ice for water. Mid-latitude glaciers are also rapidly retreating. So as I mentioned, we're in this deglaciation event, and I can show you some pictures. I won't show you data, but I can show you some pictures that indicate that. And then sort of important for our talk today is snow and ice microbes lower albedo. So they accelerate melt and contribute to glacial retreat. And so again, we're interested in what controls their distribution. And then eventually we'd love to link that distribution to albedo and their impacts on deglaciation. So probably not something I really need to harp on here in this crowd, but there are these key events in Earth's history. From the Archean, first evidence for the evolution for life. Life first evolved or emerged. Then we have the Great Oxidation Event. So this is coincident with, or sometime around the evolution of oxygenic photosynthesis. We see the first sort of appreciable amount of oxygen in the atmosphere in the world's ocean. And potentially co-occurring or just after the Great Oxidation Event, but also throughout the Proterozoic and Phanerozoic, we have these periods of major glaciations. And so the idea here from a microbiological or biological standpoint is that ice has provided a significant amount of niche space throughout Earth's history. So a lot of what we think about is sort of in this sort of terrestrial greenish realm in terms of macrofauna and where we ourselves occupy the most land space. But if we think about this is the ice extent during the last ice age, just before the last ice age collapsed, this is a significant amount of ice compared to what is present on Earth today. And I should have looked up where we are here in Washington. But normally I tell people that in my office in the Twin Cities in Minnesota, around this time, 12 to 14,000 years ago, I would have been standing under half a mile of ice. So it's a significant amount of land space, but also a significant amount of ice in general. And if we think about snowball Earth events, those are called snowball Earth events. And so we can think of these regions as being a nice place for snow to hang out for a significant period of time. If you want snow to persist into the melt season, for example, you could put it on an iceberg or an ice cube and it would last longer. So I'll just show you some evidence that deglaciations are occurring today. And throughout Earth's history, deglaciations have been linked to increasing temperature and increasing CO2. This is a picture of Collier Glacier on North Sister in Oregon in 1914. Anyone been to Collier? All right, we have one Collier. So this is a picture from 1914. This is a picture from 2011. So about 100 years later, Collier Glacier from a similar vantage point. And so for me, it's kind of hard to picture here exactly how much ice loss this is, but I'll just draw your attention to these two humans here. And so I think that hopefully gives you some scale of the amount of ice that has been lost from Collier Glacier over the past 100 years. And my joke is that I just have to keep walking farther and farther to get to my field site up here on Collier Glacier. But there is significant deglaciation, particularly here in Washington and Oregon. Basically all mid-latitude glaciers are in retreat. We can also look at evidence from years past. So this is this graph here on the left represents about 425,000 years before present. And we see in blue is temperature, so oscillations in temperature. And then in these sort of yellow dots, we see CO2. So we see of increasing temperature with increasing CO2 or decreasing temperature with decreasing CO2. And then this is evidence for, I'm out of date now. This is all the way from a couple weeks ago, April 26th, our current levels of CO2 in the atmosphere being around 422 ppm. So we are somewhere on this increasing trend, right, of increasing temperature, increasing CO2 in the atmosphere, and thus deglaciation. All right, so the first sort of little tiny thing I want to tell you about is the role of ice in supporting downstream ecosystems. And again, this is a hydrology story and not yet a microbiology story, but we really want to link this to microbes and the delivery of nutrients eventually. So glacial melt contributes to both surface runoff and mountain block recharge. This is work done by Jordan. Who's been to Collier? Yeah. So this is, I don't know, a ways up the trail, but we've got a long hike left. We're probably like 5 miles in, and this is Jordan doing a handstand. And I'm immediately wondering, like, if we should maybe put some more things in her backpack at this point in time, because I certainly was not doing a handstand at that point. Anyway, Jordan was a hydrology PhD student. She has some work. So this is a sort of a schematic of a mountain glacier where we have sort of these multiple, we have this surface, superglacial drainage, we have end glacial drainage through cracks and crevasses in the ice surface. And then a lot of what we think about in terms of water leaving the system is through superficial flow and recharging shallow aquifers. We also have a couple of other processes, sublimation and evaporation, resulting in liquid water leaving the system, and then precipitation delivering water to the system. There's also subglacial drainage that occurs at the rock ice interface. And so what we wanted to do in this study is really think about if glaciers are recharging the deeper mountain block in this case, and then how that might differ depending on the bedrock that you're in. And so for this study, Jordan collected a number of different samples from Mount Hood, in and around Mount Hood, close to the glacier, and then over 10 plus kilometers away from Mount Hood. And then she did a similar thing in Glacier National Park near a few glaciers and then downstream. So she wanted a series of streams that are both sort of, you can see maybe the glacier in sight and you can sort of assume that or think that that glacier or that stream is stemming from the glacier. And then she wanted a series of streams that were farther away where you couldn't quite tell, you wouldn't necessarily be able to assume the source. And the same thing in Glacier National Park, although it's somewhat easier in Glacier because of the distribution of the glaciers there. And so in these green dots, you can see sort of systems that are streams that are more glacially recharged. And then in the blue, we'll have the sites that are more snow or rain recharged. And so show you that data in a minute. But again, what we were interested in knowing is whether or not this glacial ice was charging the mountain block and thus supporting perennial flow at times when snow or precipitation was gone. So if you don't have this sort of snow or regular precipitation, can you still have sort of downstream ecosystems that are supported by ice, glacial ice? And then eventually we'd like to take that to the microbiology and ask how much of glacial or snow microbes are ending up downstream. Okay, so what Jordan did was measure hydrogen and oxygen, O18 in the water. And she did a series of modeling to look at where the streams and springs that she sampled fell, comparing to here in the brown, we have this rain and member zone. Here in this blue, I think that's blue. I'm a little bit colorblind, so this is snow, I'll tell you that much. This is snow. And then down here in the far left corner is her glacial ice and member, which I believe is more green. And so we see all these samples fall along this meteoric water line. But what we can see is that her end members, of course, fall here and here. But we see this sort of distribution of streams where they are all basically a mix of glacial ice, snow, and rain. If we move to Mount Hood, this is still the same isotopic measurements here on the X and the Y. Here we have an end member of snow and an end member of glacial ice. And what you can see again is that along these sort of, along this axis or between these two different end members, we see a mixture of glacial ice and snow. So these streams we see even far, far down away from glacier ice can still be a mixture of snow, glacial ice, or rain. So in 26% of the sites in Mount Hood and 54% of the sites in Glacier National Park, these streams can be primarily supported by glacial meltwater. And this glacial meltwater in this case is coming from deeper in the mountain block recharge. Mount Hood springs are supported by glacial melt recharge more than 10 kilometers from glacial ice. So you can get quite a ways away from these glacial systems and still have water from the glacial system. In Glacier National Park, the movement of glacial ice melt to recharge the springs was sort of much more constrained to a much smaller area closer to the glacier. And we think this has to do with the permeability of rock. The rocks in Glacier National Park being quite different than the stratovolcanoes you have here on Mount Hood. So this is sort of leading us to a place where we can now take, so we've got these little X's here. This is all the places where we sampled, where we sequenced microbes from the water samples. And so our next sort of place we're taking this data is to ask if those glacier microbes are ending up downstream, if glacier nutrients are ending up downstream, and what sort of downstream ecosystems they're supporting. Again, this idea that here's your glacier, but that glacier might be supporting life in a much broader area than the glacial ice itself. Okay, but you came to hear about pink snow. I know you did. And so we're interested in thinking about the potential pink snow to have contributed to past glacial cycles. And again, we're just really asking basic science questions about how do microbes live on snow and ice? Where do they get their nutrients? What are the microbes that live there? How do they interact with one another? And then eventually we'd also like to think about how they thrive on snow and ice seasonally, and what are the reservoirs for those cells from year to year. All right, so this is a pink snow field in the Beartooth Mountains. Anyone seen pink snow? More pinks? Okay, you probably don't have to go as far as Collier. Collier is quite a hike to see pink snow, lots of pink snow. This is a pink snow snowfield on the Beartooth Mountains. So the Beartooth Mountains separate Montana and Wyoming near Yellowstone National Park. And this picture is from August 2019. If you've ever sort of stood in pink snow or scraped it, you can sort of scrape it away and see this more clean or whitish snow underneath. You can put it in these highly scientific Ziploc bags and bring it back to your lab. When you melt it, it's very pink. There's a lot of pigment in these cells. But why is snow pink? That's probably the more interesting question. So pink snow hosts blooms of red pigmented algae. If we take samples of snow from many different places, we have about 250 samples in the lab now. Many of them throughout the Pacific Northwest or the Rocky Mountains. There are a lot of algae. So if we sum up all these samples and average them out, about 60-ish, 65% of the community is eukaryotic algae. So these are organisms that harvest light from the sun, photooxidize water to fuel primary productivity. Why are they pink though? They are pink because they make astaxanthin, this sort of special pigment that helps protect them from UV. So these, this snowfield is 10,000 feet. The snow that you've probably seen is probably hanging out there, mostly above tree line. Those organisms are bombarded by UV radiation on a regular basis. And it turns out, even if you're a phototroph, you love sunlight, you can harvest sunlight to fuel cellular processes, but actually most of that, or a lot of that sun is too much. And in this case, for these organisms, that sunlight is too much. And so they make this pigment as a way to sort of help them as a sunscreen to help combat UV damage. I don't know if you've seen, but there's a large, there's a whole other resource, or a whole other way in which snow algae are valuable to other communities. And so they have been marketed for their antioxidant properties. And so snow algae extracts themselves are marketed as and facial creams and other things for there. Anyway, that's not what we study in my lab, but you might see these on the market sometimes, and they are quite expensive. One thing that happens in the lab sometimes, so these algae are taxonomically, they're green algae. They're only pink because they make these special pigments to help them as their sunscreen. And so sometimes in the lab they turn green. Taxonomically, they're sort of very similar to things like Chlamydomonas reinhardtii, which is kind of the E. coli of the algal world. And so there's a lot of sort of intergenus and species level diversity. But at the end of the day, in terms of sort of microbial diversity, and we think about how diverse microbes are, snow algae are not necessarily that diverse, but are globally distributed. We've got a hole here to fill, and we can fill that mainly with fungi in terms of the eukaryotic population. So again, about 65% of those communities are almost always algae. Fungi make up the most of the rest of that community. And then in terms of bacteria and archaea, we see heterotrophs. So these are organisms that rely on organic carbon. They can't fix their own CO2 or inorganic carbon into biomass. So we've got this algae-dominated system. So algae are presumably supporting the system through their phototrophic activity. They're fixing CO2 into organic carbon. And then we've got these sort of heterotrophs, fungi, and bacteria and archaea hanging out with them. And so snow is host to diverse assemblages of algae, fungi, bacteria, and some small percentage of archaea. Where is pink snow? Where's everyone seen pink snow here? On glaciers, snow on glaciers. Yeah, so pink snow can occur on basically any snowfield that persists into the melt season. So like essentially all life, snow algae and snow microbes require liquid water to grow. And so they typically will bloom later into the season once liquid water is available. And when they're not, because they rely on phototrophy or photosynthesis or sunlight, they're not necessarily surviving in places where they're regularly buried in snow. So this is a distribution of snow just a couple months ago in March 2022. And so this, you see these sort of white 100%, and then as we get more into the blue, that's slightly less snow cover. And if we look at August 2021 in comparison, you see these sort of blue ranges here, sort of more representative of where we might think about finding snow algae close in our proximity here. So this in sort of late May is probably when the snow algae is going to start to bloom around here in the Rockies or in Montana. Usually that is happening a little bit later into the system, into the melt season. So typically where we see snow algae in the Beartooth Pass, for example, is around late July or August. So anyway, the idea being that these organisms need liquid water to grow. A good place to find them, at least around here, is on top of glaciers, where they're hanging out on ice cubes so that snow can persist longer. All right. Snow algae do a thing that is kind of interesting and kind of terrible at the same time. They lower albedo and they accelerate melts and contribute to glacial retreat. So I've been showing you these dark slides, and I just wanted to show this white slide and have you sort of picture in your mind, if you can, hopefully many, many months from now, a brand new fresh snowfall on a very bright sunny day. It's almost blinding, right? It's really hard to look at. Snow, fresh, clean snow has the highest albedo of any natural substance. So nearly all of that radiation, nearly all of that sunlight that reaches snow bounces right back off. Anything else on the snow makes that snow absorb more of that energy, more of that heat, and accelerates melt. So here we've got our, I don't know what happened to our, we've got our clean snowflake here and our clean snow. And then in comparison, we have sort of this debris-rich snow. And so this debris-rich snow is going to absorb more of that UV rate, more of that sunlight, more of that, and dissipate that as heat. So this dirty or debris-rich snow is going to melt a lot more quickly than our clean snow. Snow algae, as you've seen, and I showed you pictures of, it looks somewhat similar, right? So we don't have our clean snow anymore. We have this highly pigmented snow algae blooming on the snow surface. And so that just by being pigmented itself lowers the albedo. Snow algae also do something where they sort of increase the liquid water around them by dissipating spare photosynthetic light, if you will, as heat. And so they are both increasing the meltwater around them through biological activity, but also simply by being this bright pink color. Okay, so let's tell you something interesting. I'm going to tell you that snow microbes, or show you data, that snow microbes sequester elements from local bedrock, and that snow microbes rely on external delivery of nutrients. I should go back to this clean white snow, for example, for reference again. And this snow is not high in nutrients, at least in terms of what we think about what biology needs. We can bring this back to the lab and we can ask if there's nitrate, if there's ammonium. We can look for phosphorus or phosphate. Normally it's all below detection limits or very, very low. And so this sort of thinking about snow, that clean snow in comparison to what we see here in this sort of bright pink, dense snow algal bloom, is there's some sort of, something has to be amiss there, right? To be able to support this amount of life in a sort of very nutrient or oligotrophic medium. And so I'm going to show you that they snow microbes and snow algae, and specifically potentially sequester elements from local bedrock, so that local bedrock being a source of nutrients. And then I'm going to show you that snow microbes rely on delivery of nutrients, of external nutrients. And so this, again, is really just basic questions about how snow microbes make a living, but we really sort of need to understand these things to understand the extent of the potential for these microbes to have been around during different snowball Earth events and their potential role in biogeochemical cycling during that time. So this is just a sort of cartoony schematic where we've got snow algae here on the top of our snow, hanging out on our glacier. We have a number of different ways for water to move through the system, as I've shown you previously. But then we have ways for nutrients to be delivered, potentially through aeolian transport and potentially through precipitation to the superglacial surface. And so I just wanted to show you this picture from Mount Hood. This is looking up at Elliott Glacier. And then if you look down the valley, sort of this moraine valley, if you will, from Elliott Glacier, this is just a lot of loose, unconsolidated rock flour from the movement of the glacier, having ground up bedrock. And here we are walking out from Elliott Glacier. And so there's a lot of things near this surface, near this glacier, Elliott Glacier, that can end up being a lot of unconsolidated sediments, et cetera, inorganic stuff that can end up on the glacier. There's also, depending on where you are, there might be small shrubs or grasses in this region. There can also be things like wildfire ash and other sources of organic nutrients. And then, oh, I did put it in. I mentioned this earlier, but snow and ice are nutrient poor, not much in snow. So for this part of the study, we focused on Gotcha Glacier on Mount Adams. We focused on Elliott Glacier on Mount Hood, and then we sampled a couple of these glaciers, Collier Glacier and Dillard Glacier on the Sisters, which are near Bend, Oregon. These are just some of the pictures of what those might look like. Again, this is Elliott, this is Dillard. So it's kind of coming up to Dillard, and you can see this sort of unconsolidated rock flour here. And this is Elliott again, Gotchin, and then Collier. All right, so I'm going to show you some data that suggests snow microbes sequester elements from local bedrock. And these are these data are gonna look like this. So we're gonna have magnesium to aluminum ratios, and then we're gonna change the ratio here. Here we're gonna look at iron and aluminum. And then we're going to plot some lavas. So some of that sort of rock flour that is near the glacier. And then we're going to look at the algal biomass itself. And so we're going to look at iron specifically first, because phototrophs, it turns out, need a lot of iron in the photosystems and in some of these other, in supporting proteins that support photosynthesis. These organisms require a lot of iron. So if we look at the lavas themselves, we've got atoms in the circles, North Sister in the squares, and then hood lavas in the diamonds. We see a distribution of magnesium to aluminum and iron to aluminum that looks like this. If we plot our snow algae samples, they all fall above our lava samples. And so this is biomass. So this is cells that we've collected on a filter and brought back to the lab, washed away any sort of remaining inorganic stuff, and then looked at the biomass. And so in this case, all of the biomass, the algal cells themselves, are enriched compared to the lavas. So this suggests to us that iron is being harvested from the lavas and ending up in biomass. We do this for things like phosphorus. So again, these are all magnesium to aluminum ratios. We do this for phosphorus. We see the same sort of distribution with this one North Sister algae being weird. We do manganese. So manganese is part of the photosynthesis, part of photosystem 2, a key part of the photosystem 2 reaction center, where oxygen gets photooxidized. So we see the algae are also enriched in manganese compared to the local lavas. And then we just don't see much of a relationship with calcium. And I think it's because these organisms potentially don't require calcium. Yeah.
So you have the lava, the algae, but no snow. Is that plot on there somewhere?
So this is solids. It's all solids. That's a good question, though. I don't have the, I have soluble trace element data, but I'm not going to show it today, but I can share it with you if you want. But yeah, so this is all in solids. Now I'm thinking about that data and how much of it is above detection limits. Anyway, I'm happy to talk about that later. Sorry. So all in solids, we see that snow algae are plotting above the local, sort of the local rock flour. We also wanted to think about what would happen if we added nutrients to the system. So we don't, as I mentioned, in clean snow, we don't see ammonia. We don't observe nitrate. Typically, we don't see phosphate. And so we just thought we would do sort of, this is sort of typical in biology, to do a fertilization experiment where you just add these nutrients. You add these in an aqueous form and ask what happens to, in this case, we're going to ask primary productivity, because we're interested in our snow algae, which are, again, using sunlight to fuel primary productivity. And so we're going to look at micrograms of carbon in biomass per hour. And the way that we do this is we add inorganic carbon that is heavy in 13C. And so then we just ask after a certain period of time if that 13C has ended up in biomass. So we did this at a series of glaciers, Elliott, Gotchen, and Collier. We do a treatment in the dark. Here are the dark bars, and we don't see a lot of chemoautotrophy. So it seems like whatever is fueling the system, as I mentioned before, is photosynthetic. Our yellow here are our light. So we haven't added anything except inorganic carbon, and we see that these organisms are active. They are photosynthesizing. They're using sunlight to incorporate that inorganic carbon into biomass. When we add ammonia, nitrate, and phosphate, we see scrambled results across the board. We saw no significant increase in primary productivity, except for in this one case, a Collier glacier when we added nitrate. And then we just did a mixed bag Collier where we said, fine, let's just see if they're co-limited in ammonia, in fixed nitrogen and phosphate. And we did not simulate primary productivity. So this is like, shrug emoji. What's going on here? So they're photosynthetic, but across there isn't sort of a uniform nutrient limitation that we can identify from these results. So one other thing we did is we were interested, so I showed you these pictures of Mount Hood where you have those Elliott downstream from Elliott Glacier where you get all those unconsolidated sediments. And then I said there might be other things that end up on the snow surface. And so my partner is always, I'm always, like collecting the microbes, really sort of doing what I'm doing. And he's just off like collecting things. And I'm like, what are you doing? And he's context. He always says, Trinity, context is important. And so he's out sort of collecting different different types of microbes that we see that might be a source of food. He's collecting ice worms, which is a story for a different time. Insects, plants, needles, and soil, other things that might end up on the snow and ice surface, to ask what that looks like in terms of biomass, and if that tells us anything about where these organisms are getting nitrogen. So I have delta and 15 in biomass. Again, this is a solid measurement, and then 13C of biomass along the X. And we just sort of see this broad distribution of different stuff that ends up on or near the snow environment. And our snow algae samples themselves, all here in the red, sort of plot in this, shoot, what happened to my gray? Can you see a gray bar there? No. Let me back up for a second. So this is our snow algae sample here. If we were looking at biomass that was relying on biological nitrogen fixation, so nitrogen fixation that was catalyzed through microbial processes, we would expect to see something around 2 + 2 to -2, somewhere in this area. Our snow algae samples all fall below that, and actually they match local precipitation values. And so, as I mentioned, snow microbes rely on external delivery, and it looks like in terms of a source of fixed nitrogen, these in this region, in this cascade region, it seems like precipitation is a reliable source of fixed nitrogen for these microbes, and that is sort of not entirely unexpected. Since the Industrial Revolution, we've been pumping all sorts of nitrogen into the atmosphere, and that sort of comes back as precipitation or in precipitation. And we see gradients across the US in exactly how much, but it seems to be enough to support our snow algae populations. All right, so coming back to this not maybe great news about deglaciations being linked to temperature and CO2 and our beloved Colorado Glacier disappearing, we wanted to think about CO2 in our systems. And it's well known in many plant systems and other algal systems that CO2 stimulates primary productivity. We get larger plants in systems that are where CO2 is increased, for example. And so what I'm showing you here is dissolved inorganic carbon. So finally, some aqueous data, but not trace metals. This is dissolved inorganic carbon in water. And this dashed line here is water that's equilibrated with the atmosphere at each of these locations. And again, I'm showing you data from Gotch, Elliott, Collar, and Diller glaciers here. In this sort of first panel is the supraglacial flow. So that's as near to the glacier as we could get. And we see that in all cases except for Diller, that snow or that meltwater is undersaturated with respect to CO2. Sorry, this is supraglacial snow. This is the outwash. Either way, both of these are undersaturated with respect to CO2. I think this is fairly well accepted that snow and ice are low in CO2. If we go to sort of a final outwash, if you remember that that picture I showed of Collier Glacier, I can go back to it. This sort of would be where we would sample as a final outwash. So some of the glacier ice is now covered by debris here, but we would sample this sort of pond or glacial lake. And we see those samples still, as we're getting farther and farther away from the glacier, are undersaturated with respect to CO2. In fact, you have to get quite a ways downstream before you see any CO2 levels that are saturated with respect to CO2. So in short, snow and ice in our systems is undersaturated with respect to CO2. And this begs the question, what's going to happen with increasing atmospheric CO2, since we know that deglaciations are linked to increasing atmospheric CO2 and temperature and increasing temperature. So we did this sort of very similar experiment to what I showed you before, except here our fertilizer, if you will, is inorganic carbon, it's CO2. And so I'm going to show you some graphs that look exactly the same as before. So this is micrograms of carbon in biomass per hour. Again, we're using this sort of heavy 13C to trace photosynthetic activity into biomass. And we're doing this over a gradient of inorganic carbon, where we're adding, I think it was 50 to 250 micromolar inorganic carbon. And we're asking what happens to biomass, what happens to rates of photosynthesis. And perhaps not surprisingly, but not necessarily great news. Again, we have our dark control, and then we have over our gradient of inorganic carbon at Elliott Glacier, we see in all treatments an increase in photosynthesis as we add CO2. Inorganic carbon, I should say. And this is true across the board. So with Gotcha Glacier, we also saw an increase, and then at Collier Glacier, we also saw an increase. So an increase in primary productivity with the addition of inorganic carbon. And so we're using productivity here sort of as a way to suggest that in productivity would probably be linked to cell abundance. So as a sort of, if we allow me to extrapolate a little bit, increased productivity, increased cell abundance, increased impact on albedo. And so this sort of observation and many others has sort of led to this, a couple of popular science articles, one of them being why the last snow on Earth may be red. So again, these are organisms simply by being pink, by being pigmented and on the snow surface are lowering albedo. And yet we don't understand exactly what controls their distribution and abundance. And then I'll just remind you that what I'm talking about today maybe has a lot of us thinking about climate change today. And thinking about increasing CO2 in our atmosphere. But again, this links back to these past glaciation, deglaciation events, which are linked to increasing CO2. And really, this idea that these organisms are relying so heavily on external nutrients, we can think a lot about what might have limited them and their distribution in throughout Earth's history on these systems. Okay, so hopefully I've told you that glacial melt contributes to mountain block recharge. And again, this is thinking about supporting ecosystems farther and farther away from glaciers. I showed you some data that microbes sequester elements from local bedrock, and that snow microbes rely on external delivery of nutrients. And so it's this delivery of nutrients that probably constrains their abundance and thus their impact on albedo. And so with that, I'll end and I'll just say thanks to everyone again in the lab. And thank you. This has been super, super fun again to be somewhere in person chatting in 3D.
Hi, really nice talk. So I have a questions about your thoughts on how the algae can extract the metal elements from local black rock. And I guess my second question kind of related, have you considered the metal delivery through mineral dust in the atmosphere? It is also a delivery mechanism for other phytoplankton like a marine environment.
Yeah, so we haven't considered it a lot atmospheric deposition, but we definitely should. We thought about putting up these like air samplers to get a better sense on that. And then something else we probably should consider in terms of atmospheric deposition is dissolved organic carbon, which I think is also probably a potential source of nutrients. But your first question was about
the local black rock.
How
does the algae like extract the metal elements from the black rock?
Yes, so our prevailing hypothesis is, and it's not entirely unexpected, is that they are working with fungi. So fungi and algae probably do this a little bit too, release a lot of extracellular enzymes to do this. And so they are known to harvest things from solid inorganic substrates, but also organic substrates like wood and other things. And so our sort of our idea, our working hypothesis is that they're, exchanging carbon or something else with the fungi on the surface, and or fungi are sort of making these extracellular enzymes as common goods, and the algae are getting them that way. The algae themselves could also, make some extracellular enzymes that help in the breakdown of these nutrients or these inorganic or the solid substrates. But there's a couple of work groups that are working on looking at the snow algae and then snow fungi, and then together how those two work on solid substrates.
Are we talking about bedrock mixed in the snow, or how do they access it?
I think what you're talking about and what I was talking about is bedrock being rock flour.
Yeah.
In that case, so not the sort of bedrock, but the ground up bedrock. That's what I was thinking. Is that what you're thinking?
That's new to me, but I think it makes sense, like probably.
So I'm completely new to the fungus area of research. The idea is that the fungi, more so than the bacteria, are capable of making like siderophores and other potential enzymes that are acquiring these nutrients.
I mean, I'm sure the bacteria are as well.
I guess I was wondering, is the other group that's tackling the like fungi, algae thing because they're really a fungi group? Or is it because they believe they outcompete the bacteria for whatever nutrient acquisition and passing it off to the?
Yeah, I think either are reasonable, reasonable hypotheses or reasonable ideas. Yeah, I guess I should have mentioned the bacteria too. Just the studies that I know of are specifically looking at fungi, and because fungi are known to do this in so many other systems. But I'm sure the bacteria are. I'm sure you're absolutely right. It makes siderophores and other things that would eventually solubilize and or make those minerals available to other things in the system. Yeah.
I have another question.
Yeah, keep going.
So you have like community information. Is it based off 16S or is it a genome? Like, can you look in the community and figure out who's got more enzymes involved in?
Yeah, so what I showed you was 18S and 16S. And so we've just started doing more metagenomic sequencing to get, so I mean, the databases aren't great, but we should be able to tell whether or not it's a fungal or a bacterial enzyme. So I'm behind, as I was mentioning to someone earlier today. It's on my list to sort of look more specifically at those. And I think there's been at least one other group in I want to say British Columbia that's published a paper recently that sort of looked at something similar. But.
You made one small comment that was surprising to me and interesting.
Just one comment.
One specific one I want to ask you about, which was most of the photosynthetic organisms are algae, and the bacteria and archaea are heterotrophs.
Mhm.
And that was surprising to me, but I don't know if that's common for other communities besides just snow. What were your thoughts about that? Was that surprising to you? Or
Yeah, I mean, this is an embarrassing story actually, but so most of my work, as Sebastian mentioned, has focused on bacterial phototrophs. And so when I started my lab, I thought and nitrogen fixation. So I was gonna go to glaciers and I was gonna find cyanobacteria and study cyanobacteria and nitrogen fixation on snow. It turns out that nitrogen fixation, if it does happen, the activity is not very high. And we very rarely find cyanobacteria in these ecosystems. Why that is, I think I would still love to think about why that is. But yeah, so we just don't see other phototrophs in the system or other autotrophs in general.
That's interesting. Thank you.
Yeah, go ahead.
I have a related question. One of your glaciers, and I'm sorry, I can't remember which one it was, but it was the third one on this side, did have some activity in the dark. Do you think there's a chemosynthetic organism there, or do you think that was more like we didn't quite get all the dark?
I think, so based on the 16S data, which is tenuous to apply to function, it doesn't look like there's probably any autotrophs there. So it's probably like Trinity didn't wrap the system fast enough. But I mean, it's possible that there's some sort of chemosynthetic activity there, but my gut says probably, even if it is, it's a very small amount.
The pink snow, I'm trying to imagine what it was like in Minneapolis when the ice was retreating. Was it just a pink line around the melt zone and it worked up north as the ice retreated? Because it needs water, right, in order to operate. And northern, it was too cold to have water.
Mhm.
And so if that's true, then I was wondering what effect it has on the albedo, because it was just a fringe effect at the melt zone.
Yeah, so these same algae are I should have put this study up, but they're on the Greenland ice sheet. I think snow algae contributes something like 25% of melt. And so that is depending on where you are. So that's not all at the fringe, but it depends on where melt is happening. And the same is true in sort of these systems. So those of you who have seen snow algae, I guess you know that it's not everywhere. And so that's one other reason for thinking about nutrient delivery, but also like physical interactions with the snow itself. Because we kind of get these little pools, right? Or sometimes you see these sort of pillows where we have snow algae concentrating. And very rarely do we see it, sometimes on lakes and other things, but at least the systems that I've been in, we don't see it sort of blanketing the entire glacial surface. And so you're right, I think it is not necessarily going to be distributed entirely across all of these glaciers or all of the ice sheet. It's going to be somewhat limited, but even on the Greenland ice sheet, it is sort of impacting melt on a large or a significant scale. So it just depends on, I think, where liquid water can accumulate. Yeah, I hadn't really thought about pink snow in Minnesota because we don't get any pink snow today, but I'll have to think more about what it would have looked like 12,000 years ago.
Has anyone done a study to see what if the aerial coverage of pink snow is increasing in certain areas like Greenland or the Antarctic or South America or I don't know, the Himalayas?
Mhm.
What's going on?
Yeah, I think we're just, the royal we, definitely not me, are just getting to the point where we can distinguish between algae and other stuff on the snow. And so I think we're just getting to the point with remote sensing. And so I think we're just getting to the point where we can start to do those calculations to understand. And the satellite imagery is getting better and better. The resolution, the pixel resolution is getting better and better for us. So I don't think it's necessarily been done yet. And I think there's a lot of seasonal variability on the few seasonal studies that have been done to link it to longer term trends. So I think that data is coming, but I don't think we have it yet.
I just wanted to add that I think anecdotally, especially in the Western Antarctic Peninsula, there's records of increasing snow algae that they've been noticing over time. It's like a little bit anecdotal, a little bit from like some surveys that have gone out there. But yeah, I think you're right that we're getting better.
Yeah, but we quantify it, turn into science paper.
Oh, I know that.
We'll check the Zoom question in a minute, but I want to ask my question.
Self-serving, I
Yes,
like it.
exactly. So where do you think the phosphate comes from? What do you think the phosphate source is? And then I'm really confused by those fertilization experiments, since they don't seem to be limited by either phosphate nor nitrogen. But when you give them carbon, they increase photosynthesis. So are they carbon limited, inorganic carbon limited?
So your first question is that I think phosphorus is coming from the rocks.
Okay.
I would love to know if there's another place for it to come from, but I think that's the most reasonable explanation. And then fertilization experiments. Yeah, it doesn't seem like, yeah, so that sort of the CO2 information suggests that they're not necessarily, at least immediately, limited by nitrogen or phosphorus. We've tried, and I would love to have the data for you today, but I don't quite have it. But we've done similar studies in the medicine bows in southeastern Wyoming, where we've done a full suite. I just went nuts with like a bunch of different phosphorus, nitrogen, iron, trace elements, DIC, and then the sort of the combination thereof. So individual and then a bunch of things together. And so I'm super fascinated. It's not quite exact, it's pink snow. It's not quite exactly the same system. And it's a carbonine and the rocks are slightly different, but I'm excited to see that data to see if we get at this, any of this, whether or not it's co-limitation or something else is limiting. And then I had another thought that just, I'll think about it.
Come back.
Oh, I hid everything.
Yeah. You know where to find Zoom questions.
Do I know where to find Zoom questions? Where did Zoom go? Still out there. I don't know if we're actually connected to Zoom anymore. Maybe that's why they're so quiet.
Mhm.
Anyone, Zoom, are you out there? I don't think
Yeah,
so.
we're still here.
Go ahead, Robin.
Can you read
Are
the
there
question?
questions in the comment section?
Maybe if we,
How do you know there are questions?
yeah, maybe there are
Yeah,
just are
maybe
no
there are no
questions.
questions. Okay, we're gonna assume there are no questions. Zoom people, you have 10 seconds to raise your voice if you want to ask a
I'll
question.
ask a question.
Go.
Can you hear me? Can you hear me?
Yes.
I'm worried a little, I just question here on the dispersal of the pink snow critters. You sometimes get heavy snowfalls a few years in a row and you get an ephemeral glacier that's there for a few years, may make a small rain and disappear. And closer to where you live, there were shore ice piles in Lake Superior, and to beachfront property became very valuable, and the Native Americans would use it to refrigerate, where the wind would blow all the pack ice up into like 10-meter or more high piles on the shore, and the sand from the, which Lake Superior is full of, would get on top of it, and you build a natural ice house. So places that are ephemeral but last multiple years, do they get the pink snow on them, or does it pretty much move very slowly? I'd expect the wind to move it, but I'm not sure.
Yeah, I also would expect it to be somewhat easy to move this stuff around, these critters. And they're also sort of globally distributed, and not that there's not that much diversity, which suggests that they disperse quite easily. I don't know the answer to your question. I mean, we see in the Beartooth, for example, we don't always see the same snow patches, but we see snow algae. But those are places where snow algae have already been recently. I've had one person tell me they saw snow algae in Minnesota, but they were never able to get me a sample. So I can't confirm that. But it's a really good question, and I don't know either scientifically or anecdotally or anything. I don't know the answer.
There's also a related environment. If you go out on the thick lake ice just before it melts, it will become honeycombed. And there's water like a meter, if the ice is a meter thick, there'll be pore water that forms a piece of dirt will just act as its own greenhouse and burrow right through the ice. And this must happen some on glaciers too.
Yes, yeah, yes.
So it's another environment. It's what if you we get a cold winter again, and it could be looked for right out your door.
Sure. Well, you look for it and you let me know, because I don't think I'm going to see it in Minnesota.
I'm in California, so we kind of tend not to get those things.
No, it's an excellent question. I'm not sure I know the answer.
They're just a suggestion. I didn't expect it's something as a child, you learn not to go out on the honeycombed ice because you can go through.